Good afternoon. Thank you for the opportunity. I'm going to talk about structural systems biology of Levan Biosynthesis and Bacillus subtilis. My name is Raghu, and this is my team. Darshini and Sri Lakshmi, they are undergraduate students who did part of the work, and Aruldas and U. Venkatesubramaniam are my collaborators. And I come from Sastra University, which is on the way down south in India. So there are types of microbial exopolysaccharides that we are interested in. They are either extracellularly synthesized, excreted, or that's like homopolysaccharides or heteropolysaccharides, and they are classified based on functionality, chemical structure, and molecular weight. What you see is like different types of types like dextran, palmitoylpullin, and levan. We are more interested in the levan biosynthesis, and there is a need for the cell to produce these sorry need for the cell to produce these type of exopolysaccharides. They help in the survival, biofilm formation, migration, and many things. And in specifically levan, it's a microbial exopolysaccharide which is synthesized by levan sucrase enzyme. It's produced not just by Bacillus subtilis but also by Zymomonas. There is a lot of activities of levan. There are different features that makes it very attractive for uh, industrial purposes. And there are different types of applications: feed, pharmaceuticals, food, cosmetology, and many more. And more recently it has been used in nanostructures for coating the nanocomposites and so on. Anyway, so coming to the generic engineering part of the leaven production, there are different strains of Bacillus that are where they have tried to modify different genes, and they have tried to increase the production of leaven so that it gets more economical. And in the case of Bacillus subtilis, which is of our interest, we had different types of genome scale metabolic models which have different types of advantages. So, for example, the first one that came out in 2007 has about 1,000 reactions and 988 metabolites. But there are different plus and minus about this. Like, for example, the colors on the model column represents the first genome annotation, the second genome annotation, and the green one, the third genome annotation. So there are different models that have the 2000 model for 2007 model, excuse me, uses like the 1997 genome annotation model to build, and the 2019 model uses the 1997 genome annotation model with additional enzymatic and proteomic data. So we are took this 2019 model, although that's not the most recent one, but the reason was that it had additional enzymatic and proteome data that was more carbon centric, and which is what we were interested in because we were interested in the leaven biosynthesis pathway. In the case of Bacillus subtilis, when we map the genome and we look at where these uh, genes for the Levan biosynthesis are present, they are present in four different clusters, gene clusters, and the important genes that are involved in the biosynthesis are highlighted in the boxes. So the SAC and LEV genes are depicted here, and they belong to the SAC operon, and the directionality indicates the gene they are arranged either in the 5 prime or the 3 prime direction. And when we look at the metabolic pathway of the leaven biosynthesis, they have different components. One is the extracellular, of course, and the intracellular part. We have added the glycolytic pathway as part of this metabolic pathway because that's when we were able to balance the flux and the equilibrium. So when we tried to just have the leaven sucrase part of the extracellular from the fructose, we were not able to complete the picture, and that is why we had to add the intracellular part as well. So the minimal model that's represented here the boxes correspond to metabolites. The enzymes are shown with the additional cofactors that are involved, and the membrane bound proteins of LEVD, E, F, and G, along with SACP, are also shown. So when we took this particular model, and we did the flux analysis. The shortest path we were able to identify towards growth were in the glycolysis pathway, electron transport pathway, and sucrose transferase system. What you see on the right side of the screen is the directionality of the flux, which goes away, either away from the objective function or towards the objective function. Um, so the when we map the phase plane analysis, the which shows the relationship between the sucrose and the oxygen content and its response to cellular growth, we found that the optimal value was maximal at 18 millimolar uh, to of in sucrose and oxygen at 5 millimolar. And the robustness analysis that we did was to see what's the maximum uptake of sucrose, and it was at a rate of 6. We further did a single gene knockout analysis to show that what genes were essential for the cell survival. So out of a total of 847 genes, 170 gene deletions were simulated or predicted to be toxic to cellular growth, and 33 gene mutants negatively affected the biomass. Further, taking it one step ahead to double gene knockout analysis, we did with the pairwise genes, and this shows that they are either when both genes are knocked, it leads to either cellular death or with zero biomass growth. The darker shade indicates minimum or no growth, and the lighter shade show maximum growth rate. So here we had 95, 361 knockouts apart from the single gene knockout from the previous data, and 95 additional genes that are capable of working in pairs for cell toxicity. Specifically for this, when we looked at the genes that are involved in the Levan biosynthesis, the SAC group genes were also identified, which is shown in zoomed in the picture, uh, identified as lethal when we knocked out with pairs of SAC-B, SAC-A, SAC-C, and this shows that these genes are important in cellular growth, and they take up sucrose as the sole carbon source. Yeah. So the relative flux analysis, which is masked by the very high number of genes on the flux span, but what I want you guys to focus is on the red box at the way end of the plot, which are nothing but they are showing maximal value change for the number of reactions that are happening, and these PGK and CTAD genes, when they were knocked out, the increase in the flux of Levan were about 1.3 and 1.4 fold. We further did some gene knockout analysis using OptFlux to validate what we had done with the previous data, and we found that these additional gene knockouts were identified using SPEA2 or evolutionary and simulated annealing algorithm, and they also resulted in 27 unique genes, which we consider them having uh, higher repeatability of at least 30% or more. So there were more genes that we identified that are involved in transport, metabolism, and biosynthesis. So this was this up to this area we were able to publish it in the in a paper early this year, and then we took inspiration from Palson's group where where they did the structural systems biology part, and so when we wanted to incorporate the structural data to this pathway, we incorporated the keg mapping and the uniprot mapping, and then we took the existing known PDB structures, and for those there are no structures available, we ended up taking up the AlphaFold data, and this is what the structural pathway looks like. So the figure consists of like about 30 structures, 25 from AlphaFold 2 and 5 from PDB. Apart from this, we have yeah, okay. So we looked at this, and the we mapped it back to the Azure map in E. coli, and then we found that majority of these pathway were available. Um, and we also incorporated the data from SubTVK and BioSig, BSubSig, and we developed a Boolean network of the transcriptional regulation for these pathway, and we found when we simulated it either as all true or all false, we get a different type of a gene regulatory network. So I'm just going to go to where we are right now. So we are right now trying to integrate this Boolean gene regulatory network and using trimmer to the existing model that we have. And this is where we are having some issues, and then my students are currently trying to troubleshoot it. So once we integrate this model with this gene regulatory network model with the existing model, we hope that we have a much better picture of the Levan biosynthesis. So we have conclusion. We have integrated the structures into the genome to understand how the substrate is utilized and what the product gives out. And we also have incorporated the gene regulator network using a Boolean model. And we are trying to integrate this into the existing model. And with that, I thank my collaborators and my students and the university and SysMod for the opportunity. Thank you.
Any question for our speaker? Maybe I have one. When you move to the modeling for the Boolean network, do you know the specific interaction or inhibition among all the different components that you are including, or there is also some of the interaction that you think that are important that you include in your model?
No, these are all ground truth because we took it from the BT wiki and B subsec, which are the reported empirical data. So using that, we have built this one. So they are very close to the ground truth.
Okay.
Yeah.
Okay. Thank you.